तो कोई मुझे बताएगा लास्ट क्लास में क्या हो रहा था? What is it that we were talking about in the last class? हाँ.
We were talking about fungi.
We were talking about fungi as one of the representative groups in eukaryotes, and there we saw that the fungi dimorphism पता है, dikaryon stage क्या है ये पता है, ये दोनों special features हैं fungi के, and we took a classic example of a filamentous fungi and also a unicellular fungi, both of which have an economic importance, a very strong economic importance. And we saw, I tried to introduce you to the concept of haploidy and diploidy and what happens in mitotic and meiotic cell division. ये सब कुछ take home messages थे fungi में, जो कि they are otherwise considered to be quite dull. They are not photosynthetic, so they are quite dull looking in general, other than these mushrooms and these fruiting bodies that you saw. But ये दो तीन terminologies, and the other important feature was that fungi had cell walls made up of chitin. So let me just start sharing first. Today we will talk about the next group, and this happens to be the end of the module, the second module. तो इसलिए आज तक की क्लास तक जो भी होगा वो quiz में आएगा. Like I mentioned earlier, the only other group that is left is viruses, which I am going to take up when I cover bacterial genetics because of the importance of viruses in bacterial genetics. Now, Protists, this was coined for simplest animals, but it is an ambiguous, heterogeneous and divergent group. In fact, कहीं हम पढ़ रहे थे कि it is also called as the junk drawer, because anything that would not fall under any other group like fungi, plantae, animalia or other group would be thrown into basically Protista. You will see when we talk of taxonomy and classification that the boundaries for Protista were the most blurred. They are believed to be the first eukaryotes to evolve, hence the name derived from the word protos, means first. Now, unlike fungi, you will see that they form a bright world with many having photosynthetic pigments. In fact, photosynthesis happens to be one of the pivotal roles of Protista. These pigments contribute to their colors, and also you have a lot of ornamentation here, beautiful walls if you can see of different kinds of material. कोई protist, तुम लोग ने edible protist के बारे में जानते हो कोई? Anybody? ठीक है. ये ग्रीन कलर का क्या है लेफ्ट साइड में?
Ma'am, is it seaweed or agar?
Yes, it is seaweed. We consume quite a few protists and their products. For example, you know that agar and carrageenan are used for solidification in food industry in an algal product, while seaweeds are highly nutritious. India में तो उतना common नहीं है, but it's eaten in many other parts of the world. Besides their role in the food industry, they're now being used to produce even biomass. In today's world, you know of the importance of biofuels. So whether it is production of biomass or for hydrogen production, algae, especially algae from the protist group, play an important role. There are several pigments which are extracted from protists. I told you they are pigmented, they are colorful. So these pigments are used as alternatives in chemicals, alternatives to chemicals in some of the industries. Then the seaweeds that we just saw, they are so nutritious that they're also applied as fertilizers in agriculture. Another important role is the diatomaceous earth. I'm not sure if you know of this, this is actually fossilized remains of some diatoms. And diatoms are photosynthesizing algae. They have a siliceous skeleton. So the diatomaceous earth has many industrial uses where it is used as a source of silica. It absorbs liquids and fats, so it has a major role to play in cleaning up spills. And again, because it is fine particled, it is used in filtration also as a mild abrasive. Another important role of protists is their application in wastewater treatment, where they remove many biological and chemical contaminants. In the environment, algae produce about half of the oxygen generated by photosynthesis on the planet, thereby maintaining the oxygen levels. So they serve as the prime producer, as they can photosynthesize, like I said. Hence, they are the crucial component of food chains, especially in the aquatic system. Terrestrial pe to plants have a more dominant role. However, in the aquatic systems, it is the protists which are the primary producers. And similar to fungi, they also associate with other organisms, especially their association with corals are quite important. Because of their versatility, I told you any and everything that could not fall under other groups have been put under protists. So they're quite versatile in terms of nutrition, in terms of structure, so on and so forth. They're involved in various processes in the environment. And here are some roles that they play in the soil. Many soil protists, they feed on bacteria and fungi, even on other protists and animals. So they affect the abundance and the diversity in nature. So population control happens to be one important aspect that they play in the ecosystem. Then some soil protists, they contribute to organic matter degradation, just like you saw the fungi. They are saprophytes. While some, they are photosynthetic. They contain chlorophyll and perform photosynthesis. So they're involved in fixing carbon, thereby enriching the soil. But together with this, there are some negative associations also. How many of you know of protists as disease-causing agents? Anybody? Or any organism that you know is a causal agent of a disease? Human disease.
Plasmodium causes malaria.
Yes, that is the best example. There are several associations which are negative with associations with human beings which are negative. For example, trichomonas leads to a sexually transmitted disease, while trypanosoma, it is trypanosoma, there's a spell check, leads to sleeping sickness. And Giardia is responsible for diarrhea, while leishmania causes leishmaniasis. So there are many more. Like you said, Plasmodium happens to be the most important one, the most common one for us. Now talking of some of the general features of protists. They are microscopic and unicellular, mostly microscopic, but there are also multicellular forms. You already saw the seaweeds, right? They may be aerobic or anaerobic. They usually prefer aquatic environments. The single cell protists, they range in micrometer to the multicellular ones scaling up to meters. And another important aspect with protists is that they are motile. But the mode of motility is quite varied. And I've here put 3 pictures of 3 different motility features. For example, it could be a ciliate, it could be a flagellate, or it could even be an amoeboid organism moving through pseudopodias. तुम लोग ने ये तीनों organism स्कूल में पढ़े हैं ना?
Yes, ma'am.
Yes, ma'am.
Yes. Now there's a range of nutritional types also in protists. Some are photoautotrophs and are characterized by the presence of chloroplasts, while others are heterotrophic. Photoautotrophs matlab using light to make their own food. Heterotrophs is when they consume organic materials which are prepared by others, maybe the primary producers or the primary consumers, and they consume organic materials to obtain nutrition. Protists में क्या है? Some of them are even mixotrophs. Matlab, depending on the environment, they can combine photosynthesis and heterotrophic nutrition. They can switch to these 2 modes. When they are heterotrophic, matlab they take food from the outside, they can either have ingestive mode of nutrition. Example बताना, ingestive mode कहाँ होता है? कौन से organism में ingestive mode of nutrition होता है? इसकी बात हुई है हम लोग की. Ingestive mode.
मैम अमीबा.
हाँ, प्रोटिस्ट के अलावा लेकिन
ओके.
मैम
ह्यूमन बीइंग।
फंगाई के अलावा तो सब में इंजेस्ट करके फिर वो करते हैं ना, क्या कहते हैं, डाइजेस्ट.
हाँ, मतलब हायर ऑर्गेनिज्म में मोस्टली आप इंजेस्टिव ही देखोगे, तो हायर ऑर्गेनिज्म
यस.
इंक्लूड्स एनी ऑफ द एनिमल्स इंक्लूडिंग ह्यूमन बीइंग्स, ठीक है। और इट कुड इवन बी अब्जॉर्प्टिव, तो हम तो आपको रेंज बता रहे हैं कि ये अक्रॉस द मेटाबॉलिक डाइवर्सिटी यू विल हैव प्रोटिस्ट हैविंग वन और द अदर मोड, सो इवन इंजेस्टिव इज फाउंड इफ यू रिमेंबर। absorptive तो उन्होंने फंजाई में देखा ही था. What you see in the figure on the right is actually the process of phagocytosis, in which the cell membrane it engulfs a food particle, जैसा तुमने अमीबा में देखा हुआ है, and it draws it inward, pinching off an intracellular membrane sac or a vesicle called as the food vacuole, जिसको कि phagosome भी कहते हैं, phagocytosis इसलिए phagosome. Now, this phagosome fuses with a lysosome, which contains hydrolytic enzymes to produce a phagolysosome. And the food particle is broken down into smaller molecules, which can diffuse into the cytoplasm and be used in cellular metabolism. And the undigested material remains, which are in the vesicle, they are expelled from the cell via exocytosis. So basically, this is endocytosis taking into the cell, and exocytosis is throwing out of the cell. And this is just a real life imaging of the process of food intake by amoeba, जो तुमने already पढ़ा है कि pseudopodia extend करके food particle को लेता है. So phagocytosis तुमने अभी तक नहीं देखा था, प्रोकैरियोट्स में phagocytosis नहीं होता है, and protista में ये बहुत common है. इनफैक्ट इससे अगर तुम्हें मेम्ब्रेन की फ्लेक्सिबिलिटी भी समझ में आनी चाहिए कि कैसे ये फ्यूज करके कैसे वेसिकल्स पिंच ऑफ होते हैं और कैसे ये एक्सपेल होते हैं, जो तुमने एंडोप्लाज्मिक रेटिकुलम और गोल्गी बॉडी के कंटेक्स्ट में भी देखा था. Let's look at the morphology of protists. The cells of protists are among the most elaborate of all cells. Especially, like I mentioned, the cell walls are quite ornamented. They're colored organisms. Some cells may be enveloped simply by cell membranes, just like animal cells, while others have cell walls. Some have thin-layered proteinaceous covering called as pellicle. This is a term restricted to protists, and where they have only a thin layer of support to the plasma membrane. In protists, the vacuoles are of much significance. So vacuoles, eukaryotes they serve the function of storage. But besides that, in protists, there are special osmoregulatory vacuoles called as contractile vacuoles. Now, because of their aquatic habitat, they generally prefer these vacuoles which expel water from the cells, especially those which do not have a rigid cell wall and they have a free influx of water. For them, these vacuoles are crucial. Another unique feature in ciliates is that these single-celled eukaryotes, they have two nuclei which have completely different genomes, completely different genetic material. So the smaller of the two, which is called the micronucleus, contains the germline genome. This is the genetic information which is passed on to the progeny at the next sexual generation, and it's usually during normal growth, it is silent. There is not much role to play. But the larger macronucleus, also called as the somatic nucleus, performs somatic functions. So this is the one which is responsible for gene expression and determines the cell's phenotype, how it looks like, what it performs, the metabolic and development functions. So this is also typically found in some ciliate protists. You have seen this organism. I have selected this only to show you that it has most of the features that we just talked about. So this is a paramecium, which has a shape resembling the sole of a shoe and is a freshwater organism. Its whole body is covered with cilia, these hair-like filaments, which help not just in locomotion, but they also help in bringing or dragging the food to its oral cavity, from which it takes in the food, ingests the food. The cell is covered with a flexible, thin membrane, like I said, outside the plasma membrane, called as the pellicle. And you can notice that here you have both the contractile vacuoles as well as the food vacuoles. Then there is an anal pore or cytoproct from which the waste is excreted out. This is just to show you how it moves in real life. So if you see the cilia, unlike the flagella, make such kind of a wavy movement. So this undulating movement is what helps the paramecium move. The next organism about which I'll talk a little in detail, so iska I will take up the life cycle as well. These oval cells are narrow at the anterior end, if you can see towards the top. There is a smooth and thin but firm cell wall made up of cellulose. Cellulose आपको plant में मिलता है या फिर green alga में. This is a green algae. Besides the typical structure of a eukaryotic cell, you will notice a single large chloroplast. So you see most of the cell volume is actually taken up by this chloroplast, this large chloroplast, which could have one or several pyrenoids. Now, pyrenoids are again unique. They are sites concerned with synthesis of starch. So basically starch is the storage molecule here, polysaccharide. And in the chloroplast, when carbon dioxide is fixed, it is stored in the form of starch and the structures are called pyrenoids. There are two flagella anteriorly located. Each of the flagella, as is evident, originates from a basal body. We already saw the difference between the flagella of prokaryotes and that of eukaryotes. किसी को याद है? क्या difference था? दोनों में flagella होता है. Prokaryotes और eukaryotes में क्या difference होता है? Prokaryotic flagella और eukaryotic flagella में? Anybody? What is it made up of? What is the prokaryotic flagella made up of compared to a eukaryotic? Compare it in terms of its subunits or structure. बोलो भाई कोई. हाँ विशंतन. विशंतन अनम्यूट करो. क्या डिफरेंस था प्रोकैरियोटिक यूकैरियोटिक फ्लैजेला में? विशंतन आई कांट हियर यू. फिर से अनम्यूट करो क्योंकि बैकग्राउंड आवाज आ रही थी तुम्हारी. Anybody else who would like to answer? क्या था? What are they made up of? What is the prokaryotic flagella made up of? हाँ आशी.
Ma'am, the eukaryotic one is made up of tubulin, that is microtubulins and
Prokaryotic?
ma'am, prokaryotic.
Flagellin सबसे easy है वो. Flagellin.
Yeah, right.
Okay. और स्ट्रक्चर में क्या डिफरेंस था? कैसे अरेंज थे ये प्रोटीन्स? बहुत कैरेक्टरिस्टिक है ये. अगर तुम्हें याद हो प्रोकैरियोटिक में हेलिकली. हाँ रितिका, टेल मी.
Ma'am, in eukaryotic, I remember that there was a 9+2 system.
Correct. Yes, those
जो present in
microtubules
the
are.
prokaryote.
हाँ हाँ. Prokaryotes में बहुत simple structure होता है. It's a helically arranged. These flagellin proteins are helically arranged. Okay. While in eukaryotes you have a more complex arrangement. जो transverse section हम तुम्हें दिखा रहे थे, wherein 9+2 और 9+0 arrangement दिखा रहे थे. तो बेसिकली फ्लैजेला सिर्फ नाम कॉमन है दोनों में और दोनों लोकोमोशन के लिए यूज होते हैं, लेकिन सुपरफिशियल रिजेम्बलेंस है both in terms of the subunits that it has and the structure. ठीक है. विशंतन, अब you can lower your hands because we'll move on. तो इसमें यूकैरियोटिक फ्लैजेला है और बेसल बॉडी है. अगर तुम्हें याद हो हमने बेसल बॉडी का बताया था कि बेसल बॉडी में 9+0 अरेंजमेंट होता है, सेंट्रल पेयर मिसिंग होता है, वाइल फ्लैजेला का जो फिलामेंट है उसमें 9+2 अरेंजमेंट होता है ऑफ माइक्रोट्यूब्यूल्स, एंड माइक्रोट्यूब्यूल्स आर साइटोस्केलेटल एलिमेंट्स. ये भी मैंने तुम लोग को बताया था. Now each cell contains two contractile vacuoles located at the base of the flagella, and like I said, they help in osmoregulation. एक और feature है, the species, most of these Chlamydomonas species, they have a orange to reddish carotene containing, carotene is a pigment containing eye spot or stigma. Now this is a photoreceptive organ. For locomotion it is receptive to light. So these help in photoreception. Okay, coming to the life cycle of Chlamydomonas. It's a eukaryote, so it reproduces both asexually and sexually. Asexually is by the formation of these kinds of spores called as zoospores. Now, zoospores जब भी हम कहते हैं ना, they are motile spores. Zoo माने motile animals, zoology. So these zoospores are flagellated, or non-motile structures could also be formed. Now, non-motile structures are formed in asexual reproduction in protists when there is some unfavorable conditions. So the non-motile structures could be, for example, aplanospores without these flagella. And if the conditions are still harsher or extremely unfavorable, then you have something called as hypnospores. Now, extremely unfavorable hai, to hypnospores, they have, besides being non-motile, they have thick cell walls. The aplanospores and the hypnospores, they germinate when the conditions are favorable. Zoospores to favorable conditions में ही produce होते हैं, so they immediately germinate. Now, with respect to sexual reproduction, like I mentioned for fungi, it takes place when there is some kind of unfavorable condition. Students, मैंने microbes के लिए कहा है तुम लोगों को कि sexual reproduction में वो तभी invest करते हैं जब कोई unfavorable condition हो. And इसमें ऐसे stages होते हैं जो dormant होते हैं, which can evade stresses. So just like you had spores in fungi, here you have these resistant structures are called as CYSTS, which are resting stages. बैक्टीरिया कैसे इवेट करता है स्ट्रेस को? फंजाई में स्पोर्स होते हैं, बैक्टीरिया में क्या होते हैं? प्रोटिस्ट में सिस्ट्स होते हैं. बैक्टीरिया में क्या होता है? जो डॉर्मेंट स्ट्रक्चर्स हो या रेस्टिंग स्ट्रक्चर्स हो which can evade stress, which are quite resistant to stresses. हाँ आशी?
मैम एंडोस्पोर्स.
एंडोस्पोर्स. स्पोर्स और एंडोस्पोर्स में डिफरेंस है, ठीक है? एंडोस्पोर्स सेल के अंदर डेवलप होते हैं. हम लोगों ने देखा था मदर सेल और कैसे एसिमेट्रिकल डिवीजन होता है, और एंडोस्पोर्स आर फॉर्म दीज पीजी लेयर्स एंड द वेरियस कॉर्टेक्स एंड थिंग्स दैट वी सॉ. तो हर ऑर्गेनिज्म में एक सर्वाइव करने के लिए डॉर्मेंसी होती है, जो कि तुम हायर ऑर्गेनिजम्स में नहीं देखोगे या रेयरली देखोगे कि कोई डोमिनेंस या हाइबरनेशन टाइप एक प्रोसेस हो सकता है. अदरवाइज दिस इज नॉट देयर. सो दे हैव दिस बेसिक स्ट्रेटजी ऑफ इवेडिंग स्ट्रेसेस. तो सेक्सुअल रिप्रोडक्शन में क्या होता है? देयर आर टू गैमीट्स. अब तुम एन और 2n जानते हो क्या होता है? हाँ. सो टू गैमीट्स दे फ्यूज बाय देयर एंटीरियर एंड्स. There are different kinds of sexual reproduction. I'm not going into all those details. I'm giving you the basic idea of what happens. So then the cell wall degrades, and what is formed is a quadriflagellate cell, a cell with 4 flagella, and it also has double the cellular content. So there will be like 2 pyrenoids, there will be 2 eye spots, but eventually the flagella are lost, and the zygote, it secretes a thick wall around itself. Zygospore is formed. And it is only when the favorable conditions reappear, the zygospore germinates. And germinates का मतलब क्या है? Its diploid nucleus divides by meiosis. तुम्हें अगर याद हो तो meiosis was the reductional division. So this 2n results into 4n nuclei. And the 4 daughter zoospores, again because they have flagella, you call them zoospores. They form the vegetative cell, bringing this entire cycle to completing the entire cycle. Now, having told you about Chlamydomonas, or this green algae, there is an interesting group of organisms which I wanted to briefly talk about, which now falls under protists, but they were earlier believed to be fungi, and these are called as slime molds. Slime molds से तुम समझ सकते हो इसका appearance कैसा होता है. They do not have a cell wall in the vegetative phase, but the spores have a cellulosic cell wall, ठीक है, जो कि protists से common है. But what brings it closer to the fungi is that during reproduction, they form typical sporangium like fungi. Sporangium क्या था? Fungi में sporangium किसको कहते हैं? देव प्रिया, ध्वनि बोलो.
Ma'am, sac-like structure that
Contains
spores.
spores. Correct. So sporangium तुमने typically देखा था fungi में जहाँ पे कि it was a spore bearing structure, a sac like structure. This is present in protists. Sorry, slime molds. Slime molds are a part of protists. Okay. Then their mode of nutrition is also similar to fungi in being saprotrophic. That is, they live on dead and decaying matter. And they prefer dark and moist environments. ये भी तुमने fungi में देखा था कि moist environments, dark and slightly acidic भी requirement था fungi का. But the difference with fungi is that fungi का majorly haploid ही होती है life cycle में या diploid ही होती है? Fungi majorly haploids होते हैं या diploid students? आशी?
Ma'am, haploids.
यहाँ पे slime molds, they are majorly diploid. And again, protist-like structure होता है कि slime molds में at any one stage they will have an amoeboid phase like the protists. And the slime molds, they are actually categorized on the basis of their life cycles into two different types. जैसे कि आपने dimorphism fungi में देखा था, mycelial as well as single-celled. Yeast and mycelial stage. Here there are plasmodial phases or there are cellular phases, cellular stages in life cycle. And when I say plasmodial, they are composed of large multinucleate cells and they move along the surfaces like an amorphous blob of slime during their feeding stage. This is one mechanism of their movement and reaching out to the food. Jabki when they are in the cellular phase, they are like the independent amoeboid cells, such irregular bodies moving in the form of amoeboid, the way amoeba moves. Now the plasmodial slime mold, it begins with a spore, which is in the presence of water, it releases a mass of cytoplasm called as swarm cell. Okay, it is propelled by flagella, so it's flagellated until it comes in contact with a surface, after which it enters into the creeping phase that you saw. And here it is amoeba-like, so it's called as mixed amoeba. Here you can notice that this is N, these are N organisms till now. These may begin feeding on bacteria and they start multiplying. Haploid जब तक है तो amoeba it can reproduce by mixed amoeba form में है तो it can reproduce by binary fission. But under any adverse condition, both swarm cells or whether they are in the mixed amoeba form, they can function as gametes and they fuse to form a zygote, which is 2N. The nucleus of the diploid cell, I said diploidy is the dominant phase here. It divides by mitosis. But the cells don't separate out, they don't divide, resulting into a large cell with first two and then many nuclei. This multinucleated stage is called plasmodium. Remember, plasmodium here is a phase. Okay. So as the plasmodium grows, you can see that it forms a structure, a kind of mycelial structure. In some species, the plasmodium can be centimeters in size and contain thousands of nuclei. All this is the diploid phase, okay? It continues to grow, feeds on various microorganisms in the rotting vegetable matter, which is the saprophytic mode of lifestyle. When environmental conditions are favorable again, the slime mold changes behavior. अभी तक ये plasmodial stage में था. It changes behavior. It transforms into such fruiting bodies. ये हमने fungi में देखा था कि ऐसा stalk-like structure which bears the sporangium and then you have this sac. इसमें they undergo meiosis and the diploidy then changes. The next phase is haploid cellular phase. Finally, the spores are released into the air, and these spores, like the original amoebae, they form the next, they start the next cycle. So here you could see why the boundaries were blurred between fungi and protists. In fact, mere time pe bhi I read slime molds as fungi, but you are reading it as protists. Now coming to the last organism or group of organism that I wanted to mention. पहले तो तुम लोग बस ये देखो और मुझे बताओ कि from all what you have studied till now, from the eukaryotic cell structure, fungi, bacteria, and protist, what do you think these are? Which group would you, I don't want the name of the organism, which group will you put these organisms into? क्या है, क्या दिख रहा है तुम लोग को इसमें? बताओ.
एल्गी कह सकते हैं.
एल्गी क्यों कह रहे हो देवप्रिया, क्यों कह रहे हो एल्गी? रीजन दो.
लाइक सेल्स हैं छोटे छोटे एंड कनेक्टेड हैं एक दूसरे से, सो लगा उस तरीके से.
क्योंकि आज हम लोग प्रोटेस्ट की बात कर रहे हैं तो जरूरी नहीं है कि ये भी प्रोटेस्ट हो. ठीक है. ओके, एनीबडी एल्स? कुछ लोग तो बिल्कुल अनम्यूट करते ही नहीं है. नमन सिंह निर्वाण, पारस चौधरी, बस मैंने नाम सुना है. प्रथम बंसल देखा है. ये लोग कहाँ हैं? क्यों नहीं पार्टिसिपेट कर रहे हैं? ये क्या दिख रहा है, बोलो. बताओ ये क्या दिख रहा है. हिमांशु अनुरागी, प्लीज अनम्यूट योरसेल्फ. I would like to know from the last 3 or 4 lectures, what have you understood? ये क्या दिख रहा है? हिमांशु अनुरागी जी नहीं है. कशिश बताओगे?
Am I audible?
Yes, you're audible. कशिश, tell me. लेकिन तुम्हें रीजन देना पड़ेगा. तुम जो भी ऑर्गेनिज्म बोलोगे. हाँ, बताओ.
Ma'am, from the black and white picture, it's look like bacteria, a colony of bacteria, I guess.
Okay.
But
Okay.
the colored picture, it shows something different, like a fungi or something like that.
Good, good, Kashish. The reason why I'm showing you this picture is for what Kashish stated. These are actinomycetes, and actinomycetes are a group of unicellular bacteria that are gram positive, and they produce spores. हम protist की बात करते करते we have now moved to these bacteria for the simple reason that you can see where the boundaries are blurred here. Okay, there's special features single cells to form a mycelial structure very similar to that observed in fungi. Okay, but this is again, it's a very superficial similarity, as actinomycetes are typically prokaryotic. So there will be no compartmentalization or very simple structures. These are mostly aerobic. They are ubiquitous in nature, found in terrestrial as well as aquatic environments. The reason why I'm mentioning actinomycetes is also because they play an important role in drug development. So, how many of you know of streptomyces? सुना है?
यस मैम.
क्या, कब सुना है? क्या इम्पोर्टेंस है देवप्रिया की? स्ट्रेप्टोमाइसिस.
मैम, स्ट्रेप्टोमाइसिन एंटीबायोटिक होता है.
स्ट्रेप्टोमाइसिन, हाँ करेक्ट. तो इनका सबसे इम्पोर्टेंट रोल प्रोडक्शन ऑफ एंटीबायोटिक्स ही है. बट स्ट्रेप्टोमाइसिस द जीनस is the most economically important ones for production of a lot of secondary metabolites, especially like I said for antibiotics. These can be divided into, in fact, various classes. So for example, they secrete some regulatory compounds which include some growth factors. Then they also secrete some antagonistic compounds which can be used against, for example, protozoans or bacteria or fungi, or even antivirals they secrete. They also have an application. Some of the metabolites have an application in as agrobiologicals for controlling insects, pests, and herbicides. So they're insecticides, pesticides, and herbicides, biological agents. And then there are also pharmacological agents that are derived from streptomyces, which includes, for example, anti-tumorous compounds or enzyme inhibitors of application. And just to show you how they are similar or different to the fungi, now actinomycetes, they are quite widespread. 90% of the soil actinomycetes are actually streptomyces. They possess, इसमें एक और यूनिक फीचर है जो कि इसको डिफरेंशिएट करता है फ्रॉम प्रोकैरियोट्स, एंड streptomyces possess linear chromosome, and in fact they also possess linear plasmids. Plasmids तो हम लोगों ने हमेशा सर्कुलर देखा, वो तो यूकेरियोट्स में होते ही नहीं है, बट streptomyces में जो linear chromosome है, that is very similar to the eukaryotic genetic material. They grow at the tip of the filament. Now, one difference with the fungal hyphae is that the hyphae here is much thinner in the actinomyces compared to the fungi. The aerial filaments, like you see here, the filaments are called sporophores, and they give rise to spores. But तुमने spore का appearance देखा, जो कशिश ने बताया कि that gives an appearance of a bacterial structure wherein the spores are simply attached. So basically it's not a multicellular organism, it's a unicellular organism with hardly any connective tissue. Okay, now the difference here is that bacteria में तुमने और आशी ने बताया था शायद कि endospores होते हैं जो सेल के अंदर बनते हैं. These structures are formed inside the cell, hence called endospores, while here the spores are much like the fungal morphology wherein they are formed at the tip of the filaments. तो yeah, of course, then they disperse. यहाँ पे कोई haploidy और diploidy की बात हम नहीं कर रहे because it is all a haploid stage. This is asexual reproduction, unlike in fungi where you see these haploid, dikaryon, and diploid phases. Okay, so that brings us to the end, wherein we have discussed some of the representative organisms, starting from their structure, the morphology, the habitat, the life cycle, basically to present to you the kind of diversity that is in microbiology. In my next class, I'll talk of microbial taxonomy, post which we will concentrate majorly on bacteria. तो यहाँ तक हम eukaryotes की बात कर रहे थे, लेकिन अब we will concentrate only on bacteria. So whatever metabolism or growth or stuff we are going to study will be concentrated on prokaryotes, especially bacteria. This is just an e-book that I found. Some interesting videos that you can see, and another interesting article that I found in New Scientist. Okay, now I'm open to questions. If there is any doubt, please ask me.
मैम क्विज में पिछले लेक्चर का भी आएगा क्या?
पिछले का मतलब आज तक के सब लेक्चर का आएगा.
अच्छा,
सब कुछ, पिछले नहीं, मतलब जितना अभी तक हुआ है. पर आज के लेक्चर से कोई क्वेश्चन किसी को? ठीक है, if there are no questions, then we will see each other. जितने लोगों ने नहीं ओपन किया ना, हाँ रितिका पूछो.
मैम आई स्टिल डिडंट गेट कि वो पिक्चर से हाउ डिड वी कंक्लूड कि वो क्या है. कैन यू जस्ट एक्सप्लेन?
पिक्चर से हम बिल्कुल कंक्लूड नहीं कर पा रहे थे. मैंने तुम्हें सिर्फ कंफ्यूज करने के लिए वो पिक्चर दिखाया था, क्योंकि वो पिक्चर माइसीलियल स्ट्रक्चर था, बिल्कुल माइसीलियल स्ट्रक्चर था. ठीक है, लेकिन जहां पे तुम स्पोर्स देख रहे थे ना, उसमें तुम्हें डिस्टिंक्ट सेल्स टाइप दिख रहे थे, राइट? आपस में कोई कनेक्शन नहीं था, ये कंटिन्यूअस सेल वॉल नहीं था, जैसा कि तुम्हें फंजाई में बहुत से स्पोरेंजियम में दिखेंगे, या फिर एस्कस में दिखता था तुम्हें स्पोर्स, अगर तुम्हें याद हो, या फिर तुम बेसिडियल स्ट्रक्चर में तुम स्पोर्स देख रहे थे. तो कहीं ना कहीं इनफैक्ट अगर तुम प्लेट पे देखोगे ग्रो करके, मे बी जब प्रैक्टिकल्स होंगे तो तुम देखोगे कि फंजाई की प्लेट, अगर तुम फंजाई ग्रोइंग ऑन अ सॉलिड मीडिया देखो कंपेयर्ड टू एक्टिनोमाइसीट, देयर विल बी एब्सोल्युटली नो डिफरेंस. तो इनफैक्ट इसलिए हम तुम्हें बता रहे थे कि ये स्ट्रक्चर से कुछ नहीं पता चलता, you can have this mycelial structure which is still in unicellular and prokaryotic, and you might, we as scientists also confuse these actinomycetes as fungal structures. ठीक है, हम तुम्हें यही सिर्फ इसीलिए ये स्ट्रक्चर दिखा रहे थे कंफ्यूज करने के लिए.
Okay.
Okay. Okay then students, if there are no further